Yeah, thank you very much, Frank, Henrich. Where did you disappear? Henrich, Henrich and Margaret, for all the flowers. Yeah, I have to live up to that. Thank you also, everybody who is here in this room and who has come today. A special welcome to the students which are also here. The past 24 years I have been working in this building and in this very lecture hall. This was the inauguration lecture hall also in, I think, in June 20,000. And my predecessor, Professor, my predecessor is still here today here. That was a wonderful time. I had really fun. I'm very grateful to the faculty and to the university that I can go on a bit to work in this building, but maybe not so much in this lecture hall. And with that, in a wonderful book I read in the last few weeks, Henrich, this is a book which you also will get. I read the sentence, the book is by Eva Tind. There are two ways to study the universe. You can travel into space and risk your life, or you travel into the invisible microworld, where space is just as vast and infinite. In a way, you just turn your telescope around and look in the other direction. I'm very thankful that throughout my academic life I always could travel in this world, and I had the privilege to see the invisible and the unseen and unknown. Seeing what no one has seen before is one of the essences of being a scientist, and that makes scientists happy. And I'm infinitely grateful that throughout my life I had never to travel alone. I had always the pleasure to be with enthusiastic, energetic, younger colleagues, students, and any type of paper which we have are contributions from the many of us. And before I start, I really like to acknowledge that anything I did was always teamwork. And we have a wonderful team right now. I had wonderful teams of previous and former members, and I think many of them are, or some of them are here today. And I thank you all for that. A question I usually get, or often get, I'm not sure if Margaret gets that question. Dear Professor Bosch, why do you waste your time with Hydra and not working on something important, at least mouse? So I never learned anything in life but Hydra. I started as an undergraduate in a wonderful lab by Charlie David in Munich, and he introduced me to Hydra, a simple organism which you can culture and which has a well-defined morphology, physiology, molecular biology, very simple, 2 cell layers. And in the last 2 decades, or a little more, the molecular biology was worked out quite well. It was invented in the 18th century by a Swiss naturalist, and it was taken into reality and modern life in the year, in the 1970s, in the Max Planck in Tübingen with Alfred Gierer, as a model to understand the fundamental processes of life. There is another aspect to this animal and another architecture, and it's the architecture, as I call it, of the invisible. Modern technology has allowed us to see that this seemingly simple organism is indeed colonized with an invisible layer of microorganisms sitting outside in a kind of a mucosa type of layer which is hard to see. You need sophisticated fixation technologies. But in essence, the whole organism is covered with a mucosa-like layer where numbers of different microbes are sitting there. Numbers of them. It's diversity. It's limited diversity. And we can culture all of these bacteria. That means we can study them in depth. They interact with each other, but they also interact with the host and vice versa. That was not something we had expected. We could not foresee that this. The first was sequencing, but then you don't know where they are. But now we could see them. And the turning point of that was, as Margaret mentioned, and I think he sits in the room somewhere, Sebastian Fraune. Where is Sebastian? Oh, in the very last row, okay. Sebastian came as a graduate student with a microbiology background, and we decided to sequence Hydra species in terms of, do they have microbes, and if so, which one are there, which microbes are there? And the astonishing thing, and this was really the wow effect, as Margaret said, was that the different species had different microbes. And I really thought this is impossible, because they live, as you just saw, in plastic dishes, and I never go out in the wild, no, and not for research. And they get artificial food. How can they have different microbes? So Sebastian proved it, and we have two nice papers where we presented it. And what it meant is very clear that there is a genetic force from the host side to keep that microbe. Independent, if you live for 20 years in a plastic dish, you have your microbiome, and another species has the other microbiome. I sometimes ask how scientists find their questions. That's the most hardest thing in life. But if you have your question, I want to understand fundamental processes of life from the very beginning, as an undergraduate. And then I found my teacher, and he showed me Hydra. And I said, okay, this is a nice model. And he discouraged me to go on as a postdoc because I wouldn't find a job. And I get all my jobs because of Hydra. But when you realize that there is a genetic pressure to keep that microbiome, at that moment I made a decision, I want to understand this. I could have gone on with doing developmental biology and stem cell biology, but this was the turning point. And Sebastian was very crucial, and I'm happy that he's here. He looks a bit different these days, but he is here. There were conditions, of course, here in Kiel. When I came to Kiel, I have to tell the story. I was in Jena. I got the offer to get to Kiel. And I called my wife. She was in Munich at that time. And then I said, well, does Kiel could work out? And she immediately said, where is Kiel? And of course, we knew where Kiel is, but at the academic, scientific landscape, it was not so much on our mind. Anyway, this was in the 2000s. And so I came to Kiel. And I met Jens Schröder, and he also is here sitting somewhere. Together with Professor Christophers from the dermatology clinic. And Jens was in my lecture here in June 2000. I'm not sure if you remember. And then after the lecture, I had nothing to do with microbes. He said, Thomas, we should maybe think about epithelial defense. How do the Hydra protect themselves against the microbes? And Jens founded, and I was co-speaker, a collaborative research center. It's called Molecular Mechanisms of Epithelial Defense. It was the time when microbes were considered pathogens, when microbes were considered harmful, and then you have to understand how can you improve the defense mechanisms of particularly the human, but also the evolutionary old defense against the tissue. So it was, in biomedicine, there was one thought, that is, you have to win the war against the bacteria. And of course, you use the immune system. And having an organism like Hydra, who has only an innate immune system, I shouldn't say only, it's pretty strong. Jens asked me, let's look for epithelial defense, means antimicrobial peptides. And what we did over the years is we developed a whole scheme of from the receptors of the ligands to the signal transduction, to the transcription factors, to the target genes of the transcription factors, which are antimicrobial peptides, and they play a key role. That was a paper which had a long and a tragic history, but at the end we got it out, and it's good that it was finally out. That was an important precondition. Another precondition was, and you see Jens Schröder again, that in Kiel I immediately met a very interactive, interdisciplinary, uncomplicated community of colleagues. I was the younger one, but Stefan Schreiber and others immediately integrated me. The medical faculty integrated me, which was great. I feel every day that what we do has to have relevance for human. And so if you entered this house here, and I tell that particularly the students and the younger ones, you pass every day, you pass this plaquette there, Ort der Ideen 2006, and you think, what the hell was that? Here is Jens Schröder and Thomas Bosch and a great sponsor lady, and so we got a prize for the idea to make an interdisciplinary center, which at that time was called Center for Biochemistry and Molecular Biology. And out of this idea started then the idea we need a real building. And then Stefan Schreiber, and here I'm with the Prime Minister here, we thought about, and Margaret and Ned have seen the model over there, we thought about we need a real building, and so that's where the building is next door. And I know that Tal considers it an architectonic disaster, but we are still very proud to have that building here. So that was very important, and that was financed by the state of Schleswig-Holstein. Scientifically, a precondition for the ideas which will come now was a theory brought out by Eugen Rosenberg and his wife Ilana Silber-Rosenberg in 2008, and Eugen and his wife were several times here, and they were key in early brainstorm meetings how to make, or should we make a kind of a center on this research. And what Eugen proposed in this theory, hologenome theory of evolution, was that in addition to the genome of the host cell of your cells, which is mostly in the nucleus and a bit of that in the mitochondria, we are colonized by millions of different bacteria, and each bacterium has its own genome. Since some of these bacteria may decide to colonize the host cell with their own genomes, you have many genomes. That is the hologenome. That's one point. But maybe even more important, you cannot change your genome easily if environmental conditions change. But with your millions of bacteria, which all have their own genomes, and the bacteria can leave your body if you want, or if they want, and the new ones from the environment may enter, gives the human and the animal and the plant body, it's universal, a kind of a flexibility in the dynamics. And that is the power of the holobiont. You are more than just yourself. And the strength, and I will guide you through some of the findings, of your fitness comes from this flexible microbiome. That was Eugen Rosenberg and Ilana. This theory was super controversial. It is still controversial, and Eugen had a really hard time to put it out. I think he was very right. The holobiont concept has changed our world irrevocably. And in the very early days, Margaret and I wrote an article, Metaorganisms as the New Frontier, in 2011, long before we knew all the details, what we now knows. And another precondition, which was important, was that Margaret McFall and Mike Hadfield invited a bunch of scientists from around the world to the middle of nowhere in America, to Durham, and to the Evolutionary Synthesis Center there. And we were locked in a kind of a semi-nice hotel and in a building, and had the task, after these 3 days, we have written a paper. The paper is written, it is cited at the moment about 2,815 times. The title is Animals in a Bacterial World, a New Imperative for the Life Sciences. And it's a classic paper in the field. It makes the point, and it changed my mind again, because there was a world famous paleontologist on board, Andy Knoll. Some of you may recognize your faces here. If Uche is here, she will see her face here. And Andy Knoll, I was very proud that I'm working on a phylogenetically old, ancient organism, which is 560 million years old. And Andy told me, you know, Thomas, that is okay. But 3.5 billion years before your Hydra was there, the Earth and any kind of substratum, marine, terrestrial, aquatic, air, was full of microbes. They invented the molecular biology of life, the language of life, and then eukaryotic cells came. They became eukaryotic because of a symbiotic event, and then they developed, evolved on a biofilm of microbes. Good Christ, that means that any feature which we study in animals, plants, or humans are actually always evolved in the context of the microbiome. That was a strong group paper, thank you, Margaret, and a strong statement. And with that thinking, we came to this Holobiont Imperative, and you see now here the scheme, a modified scheme. It went through several modifications by artists of the Holobiont. And what we really mean is that while biomedicine has focused the last 150 years or longer on the host, this is not useful anymore, because there is no isolated host. The host is always associated with a myriad of prokaryotes, of viruses, of eukaryotes, and it lives in an environment, and there is lots of interactions going on, which we are studying at the moment. From that moment on, it became clear, and Philip Rosenstiel has, I think, coined the term at some point, we have to consider patients as metaorganisms. And that is very true. And you have to get to a holistic understanding and a holistic medicine in the best sense. The term made it. There are two terms which are competing a bit. I don't care, holobiont or metaorganism. But as you can see, on this graph here, you can see that it needed technological progress and breakthroughs, next generation sequencing, that people could now in different organisms really study that and analyze that. And now there is a flood of papers using that term already, and this will go on. And here in Kiel, we have contributed to that, and we have this wonderful center of this German Science Foundation, Origin and Function of Metagorganisms, which is now led by Heinrich Schulenburg. So what I want to do in the remaining time is to guide you through four aspects. I would like to show you what we think microbes do with our immune system. I would like to tell you what we think microbes do with our development. And most exciting, and I asked the student, I show unpublished data from what do microbes do with our nervous system. And when I say our, I do that, of course, very self-confident, because what we know is from Hydra. But you use Hydra as a model to get to fundamental concepts, to concepts which are valid across different species. Then you know this is something really true. And at the very end, I give you a few outlooks. So microbes, we consider them in meanwhile as the third arm of the immune system for a very good reason. If you think of the invisible architecture which we made visible, you can ask, what happens if I remove the microbes from this organism? And this is pretty dramatic. And this is another student. I see him also sitting in the last row, Søren Franzenburg. And so what we did is we removed the microbes from Hydra and got germ-free hydras. And we were very happy about that. That is not an easy task. You have to feed them sterile and you have checked that they are sterile and so on. So you had germ-free animals. But soon, Søren came in my office and said, Thomas, I have a problem. I said, what's your problem now? And he said, all my animals die. And as soon as we removed the bacteria, fungi came. And fungi got rid of the, infiltrated the animal and really killed them. And because we make germ-free animals with antibiotic, and antibiotic doesn't work for Hydra, for bacteria, so then the fungi overgrew. And it needs an interaction of different microbes to protect. Here's a fungi coming in against epithelium. Now, dear Professor Bosch, why do you waste your time with Hydra? I don't care about Hydra. I can tell you, if you go to the dentist and he tells you have parodontitis and you need oral antibiotic, and you did that a few days ago, and you took your oral antibiotic, it removed all your oral bacteria, and you look today in the mirror and you see, oh my God, that is thrush, these are yeast, this is candida growth. It's nothing nice you really want to have. It comes only because you have removed the local oral microbiome, the oral bacteria. Candida is a normal member of the oral microbiota, but if you remove the bacteria, they have lots of space, they have no competitors anymore, and they grow. So bacteria, if you conclude of that, bacteria protect yourself against this type of intruders from Hydra to the oral cavity of man. And the mechanisms that is happening in the protective side from the animal at that time, I told you, was we thought that the host cell produces antimicrobial peptides to kill the bacteria. Now that was a problem, because now we have bacteria living on the tissue, and we have lots of antimicrobial peptides, and we have really many antimicrobial peptides, and we have numbers of papers documenting that. And then another graduate student and long-term postdoc came, and I think he's also in the room, came and was focusing on one molecule which was brought to our attention as a cytokine. And René Augustin just immediately said, Thomas, that's an antimicrobial peptide. And so we isolated it and we checked it. And this antimicrobial peptide turns out that it has a very nice function because it locally shapes the microbiome along the body column. That was early days and pretty new ideas. However, with novel technologies and single-cell sequencing, and this is now work from postdoc and assistant and Lehrstuhlvertreter now Alexander Klimovic, we did a quantitative single-cell analysis and found that across all cell types in the body, cells in Hydra produce tons of antimicrobial peptides. Some of them are conserved and some of them are novel. And to make sense out of them, we found evidence and conclude that the language between the host and the microbiome in terms of biochemistry is done by antimicrobial peptides, and that is universe along the tree of life. I wrote this article together with Mike Zaslov, and I'm giving a talk, the opening talk at the Gordon Conference on antimicrobial peptides in February. Somewhere in the United States, and I gave my talk the title, A Metaorganismic Perspective of the Origin and Function of Antimicrobial Peptides. I love to talk to people, as Henry maybe said, and I love to talk to strange people like philosophers. And so when you think about that microbes are part of your immune system, that's a problem, because it contradicts one of the definitions of immune system. Immune system has to defend against non-self. Microbes, these thousands of microbes, all have their different species, they have all their genomes, they are very different organisms living their own life. They are non-self, but they are part of you. That led us, I invited two colleagues, Angela Douglas and Tobias Ries, locked them in my room here in the fourth floor and said, after this week we have written a paper, and we did that on the question, what is self in the world of the microbiome and the holobiont and the metaorganism? The second aspect I would like to briefly touch is a work which we have now submitted, and this is a work which goes back to my developmental biology history and stem cell biology, and there is an old paper in the literature in Science in '82 which says that if you remove bacteria from hydra polyps, they cannot bud anymore. That means they have to stop asexual proliferation. Wow. This is by Menachem Rahat, a very nice Israeli colleague, and Demetman, and this is strong. If microbes affect development in an ancient organism so strong, then let's find out what is behind. We and others tried, and we never could get that working. And until another graduate student, I think he's also in the room, or I'm sure he's in the room, Jinru He comes in. And Jinru then did something that nobody else did before. Jinru managed to keep germ-free animals for a long time, up to 3 years. Culturing them germ-free, sterile, feeding them, no vacation, not letting the animals give in other hands, otherwise they get contaminated. And when he did that, it doesn't take 3 years, but it takes a few weeks. If you remove the bacteria, so this is the controls, this is germ-free, and this is long-time germ-free, the animals get very weird. And if you bring back the bacteria, they get even weirder. And when Jinru showed me that, I really fall from my chair. I know hydra for 30 years, 40 years. I never saw something like that. And that's only because we removed the bacteria and gave them back. So the secret behind all that is stem cell biology and pattern formation and developmental biology, and we approach that with state-of-the-art technology. Again, single cell atlases, these gray spots and landscapes and continents are the transcriptomes of individual cell types. And in the middle is always the stem cell, and then it goes into a differentiated cell, and you can do that with all cell types of hydra. So you have a unique way of following the trajectory of differentiation from a stem cell compartment into a differentiated cell. And now you do it in presence or absence of bacteria. And Jinru discovered something extraordinary. If you look on the left side here, this is the trajectory from the stem cell, transition zone, differentiated zone. These are all thousands of different genes. But if he does the same analysis with a germ-free animal, you don't see the transition. And you see a lot of gray bars. This means these genes are not expressed at all in the absence of bacteria. And so we cannot check thousands, but we checked one of the genes which is in the stem cell compartment and which is not expressed when the bacteria are absent, and we knocked it out or down, and we could show that this gene really affects negatively the budding capacity. In conclusion, there is no budding factor provided by bacteria. That would be too much. Bacteria are living constantly on the surface of the epithelium, and they eat there, and they live there, and they have their metabolism. Scavenger, they take away something from the host cell's epithelium. And if the bacteria are not there anymore, we believe that something inhibitory is accumulating. The waste product, which normally would be taken away by the bacteria, is not taken away anymore, and this inhibitory factor or factors then influence stem cell decision making. The last aspect is truly for me at the moment the most exciting aspect. It has a history. And the history is that we started to study I'm just careful. We started to study behavior. Nobody did that before in these organisms, because behavior has a genetic background. But after what I have learned so far, I strongly believe that there probably is also a microbial influence on the behavior. And in a very first study with another graduate student, Andrea Mogillo, I'm sure she's here somewhere, she's at the moment in Max Planck in Plön. Andrea looked into one behavior of Hydra, and that is spontaneous contractile activity. And every one of you, hopefully all of your intestinal system has these contractile activities, and all organisms, you see here worms, and you see jellyfish, and you see different ones. This is a movie from Stefan Schreiber here, from humans here. I call it sometimes the pulse of life, because we did that. We learned it the hard way. If you stop that, because you want to have an animal which is really very quiet, then they die. So it's important. And I have a different paper why it is important. But what Andrea showed here is that if you remove the microbes, then the number of contractions is going drastically down. But if you give back the normal set of microbes, not individual ones, the normal set, then you rescue this spontaneous activity nearly completely. And subsequently, with a lot of molecular analysis, we showed that pacemaker cells, which are responsible for that contractile activity, have all the equipment to A, sense microbial signals, and B, talk to the microbes by releasing neuro-derived, for example, antimicrobial peptides. And again, that's true from Hydra to worms, to Drosophila, and to mice, and most likely to humans. The second aspect of this neuronal research was done by a graduate student who is now a postdoc in the Crick Institute in London. And what Christoph Gietz studied is the eating behavior. The animals have a reflex, so it goes very standardized. But there is a mouth opening, as you can wonderfully see in this animation, and this mouth opening then allows that the food comes in. And so we wondered, how is this really orchestrated at the molecular level? And in particular, what is the role of the microbiome which sits outside these animals? Now I cannot give you a full introduction to the beautiful nerve system of Hydra. At the moment I'm fighting tons of textbooks which all write it's a primitive diffuse nerve net. This is wrong. It is complicated, subpopulations, we have shown it all. You see here the beauty of the Hydra. You again look into the mouse, but now Christoph has introduced another technology, calcium imaging and transgenic neurons specific for a particular neuronal subpopulation. And each time the mouse opens, this subpopulation fires, is active, and that's why the mouse can open. So we had that we have in all the different subpopulations of the animal. I show you a scheme, and you see at the moment we work with 7 subpopulations. There may be 9 or 10, we don't know, but we work with 7, and N6 and N3 and N4, and in each one we have these transgenic markers where we can study their activity when they are active. And Christoph found that the signal from the food comes first to these N6 cells, and from the N6 cells, then it's moved to the N4 cells, and that is part of the oral nerve net here, and then it leads to mouse opening. And if we ablate means remove these cells, so we can also specifically remove these subpopulations, then the mouse opening does not work anymore. And so we knew that the two cell populations also work together, but each one, nerve 4 and nerve 6, is important. At that time, of course, we were fully the holobiont and metaorganism world, and we knew that where some bacteria are sitting, and this is the main colonizer, Curvibacter, and what you can fluorescent labeled genetically, and what you can see is this doesn't colonize the whole animal, but it's just mostly in the oral area, or very strongly in the oral area. So we can monocolonize the tissue, the animals, with just one bacterium, which is very unnatural, but very helpful for experimental reasons. And when we monocolonized them with Curvibacter, we got another surprise. And that is the fun in the lab, every day, that you get something which you were not looking for, but you got surprised. And then you have to find out what is going on. And the surprise was that when we monocolonize with Curvibacter, the animals cannot nearly open the mouth. Good Christ, that's not a good idea. Then you cannot eat, and you are certainly not fit. So why can't they open the mouth anymore? They cannot open the mouth because this bacterium produces a molecule, a metabolite, glutamate, and this glutamate, this is the N6 population here, goes to a receptor at the surface, and then it activates these cells here, and these cells here lead then finally to the inability to open the mouth. How stupid is that? There is a bacterium which produces glutamate, but then the animals cannot open the mouth anymore. And in nature, of course, animals can always open the mouth and they eat. But we did something very artificial. We monocolonized the tissue, we reduced diversity. In reality, this Curvibacter is among a community of different other bacteria. And Christoph and his team showed that, for example, there is a bacterium called Unibacterium. This Unibacterium eats and needs the glutamate. It cannot make glutamate by itself. So normally, this glutamate produced by one bacterium is never seen by the receptors because it's taken away. But if you reduce the diversity and if you remove some members of the community, this has drastic effects. From Hydra to man. Glutamate is an appetizer, and in all of your convenience food you get with Rewe and Edeka is glutamate in. The last story is maybe the most exciting one, and it's not published at all. It's done by a wonderful graduate student in the room, Christopher Noack, and we finally managed to look into embryos. I say finally because I can tell you all of this is years of working, and I have a wonderful technician. He cannot be here today, Jörg Wittlieb, who helped in that work greatly. So the animals produce embryos, and the embryos fall down, and then they hatch, and then they make a little polyp. And what we ask in this project and this study, and it is getting finished or finalized at the moment for a paper, is how do they form the nerve net de novo during embryogenesis? And Christopher introduced in the animals and into the embryos transgenic neurons, again with this calcium imager, and then followed the nerve net formation over time during the embryogenesis. And I don't go into details, but what we can follow for a subpopulation of neurons is that from an more or less unorganized situation, you come to a nerve net, which then is this nerve net for this subpopulation. That is great. And it means that the information to make the nerve net unfolds with time. Makes no sense to scribe that in the genes, which neuron has to do at what type. That doesn't really work. I think our work shows that this is a self-assembly process, and with time, a complex nerve net happens. This will be the future head region, this will be the future foot region here, with time. If you remove the bacteria, sorry, Christopher, I asked you if I can show that. It's preliminary data. If you remove the bacteria, the nerve net formation is not very successful. You see here the control. This is the embryo. There is a nerve net already with this subpopulation at the oral end, and here's the foot end. And in germ-free animals, it's very loose. And it's very loose because two reasons. There is less neurons in the germ-free animals, in the germ-free embryos. And while normally neurons mature with time and get more neurites, more appendices, in the germ-free animals, this always stays N is 2. They have a maximum of 2 neurites. This reminded me on a beautiful work by a colleague in UCLA, Elaine Zhao, and she showed in a Nature paper some years ago in the mouse system that mouse embryos developing in a mother which is kept germ-free, missing microbial metabolites, develop neurons which are much more simple and have less neurites than in the control. And the embryos are born, and the fetuses are born, and then the mouse embryos have behavior deficits in very defined manners. So it's not only that the absence of the microbes influences the nerve net formation, it's also that seems to be another universal principle. That's all what I wanted to show you today. I think the work over the last 10 years has shown that within the holobiont, there is tremendous impact on developmental biology, on immunity, and also on the nerve net. A model is a lie that helps you to understand the truth. That's why we work all on models. But it also brings you, because you work on a model, you want to think beyond the model. And I'm grateful to be a fellow of this Canadian Institute of Advanced Research, where Margaret also is a fellow. We come together about 2 or 3 times a year in the world, and we think about new frontiers in humans. I'm part of the human microbiome program. May sound very strange to you, a guy who works all life in Hydra. But so we publish on hygiene, we publish on non-communicable diseases. We were very concerned during the COVID crisis what too much hygiene means for humans. And more recently, Ned Ruby, who sits here, Margaret McFall, and Martin Blazer and myself thought, and Margaret and I had a long discussion for that for years, we have to change our language. We started with a language which was born in war. Microbes have to get killed. And Jens Schröder's SFB was there to find mechanisms how to improve epithelial defense. And that was wonderful at the time. But it's time to think that the majority of microbes are beneficial and the pathogens are a super minority. And it's time to study the rule and not the exception. And it's time to consider some of the terms. Why should we always talk about infection? It's colonization. What is a pathogen? A pathogen is context dependent. It can be harmful, yes, but in a different context. Think about the Candida experiment and your oral antibiotic. It's a normal member. It's actually useful, and there are many more. So we published this new lexicon, hoping that it has some impact. In August, I got a mail from the editor, 7th edition of the American textbook Microbiology, Marjorie Kelly Cohen. And Kelly wrote me, I have now to use my glasses, but enthusiasm about changing our language from a war footing to the idea that disease is an outcome and not an inherent characteristic of a microbe. That is something what we really want to achieve, changing textbooks. So in the very last moment here, I want to show you maybe final frontiers. I'm very sorry, I know I disappoint some, because there is a future for a senior professor. And the future of the senior professor is focused on two aspects. And one is, beside all what I told you, I'm working on an animal which is immortal. This is at least what nature has written. I don't like that. I call it non-senescent, the impossible dream. And there are hallmarks of aging. That's a famous paper. And it tells you that aging has to do with microbes, with stem cells, with nutrient sensing, and with many other things. And so one of my goals immediately, we study already that, is to understand why can this animal be non-senescent, and we all have to die. And the second aspect I'm very much intrigued is that microbes in your body and also in Hydra are not randomly distributed. They find in different locations, and we have some of them which are in the top and in the middle and in the bottom. And that's the same with you. It's on your skin, your surface of the hand has different microbiome than your palm, and that is also in the intestine. This is about 7 meters long, and you have many different microbes along. And what my dear medical molecular biologists do is they sequence the stool. This is something which comes out at the very end. But in essence, the microbes work at a given position, and nobody understands what does it mean in that position. If you change position, you get sick. I want to understand what's going on. So, in summary, making the invisible visible, I think we have a model to study that. Complexity can be reduced and then analyzed. Many processes and structures discovered appear to be universal and valid not only for Hydra, but also for vertebrates, including human beings. I showed you the example of the nervous system. And I cite our centuries-old host-centric view will inevitably fade, and development as the interplay between an animal host and a multitude of microbial prokaryotes will emerge as the rule. Too few people believe at the moment in that. There are two ways to study the universe. You can travel into space or risk your life, or you travel into the invisible microworld where space is just as vast and infinite. The last point I really want to offer, very thankful for the faculty, offering help and support to the next generation of scientists to travel into this invisible microworld, maybe more successfully than myself. I thank all the members who have contributed. I think I have mentioned the current work and the colleagues who did that, the funders, the Science Communication Lab, which is here in nearly complete. It's wonderful. And I thank in particular, I have no idea where he sits, Alex Klimovich, who is a wonderful colleague next door and takes care of so many things which I don't do. And I also thank my family, and Judith, and my wife is here, and my daughter, they are here. I'm living in a metaorganismic environment outside of Kiel, and these ponies are just in front of our garden, and I made a video for the CRC during pandemic to cheer up the students. And with that, we are nearly in time. Stay tuned, there comes more. We have a fantastic work at the moment where we replicate in vivo a complete nerve net from an asexually growing polyp. And with that, I'm very happy to meet everybody now upstairs in the reception. Thank you very much for coming.